Welcome back, fellow mitochondriacs, for another episode of Cancer as a Mitochondrial Metabolic Disease. This will make the third installment of the little mini cachexia series that I have wandered into because of lactate. And this is one of those rabbit holes that I did not really see myself going down. But now that I know that 20% of cancer-related deaths are related to cancer cachexia, this seems like a good place to pause and actually investigate to see how we can have an influence here. And the more I look into this, the more I realize that looking at cancer as a metabolic disease and using ketogenic metabolic therapy, as well as some other interventions that have been talked about multiple times on this channel, seem to be the answer, not only to the tumor itself, but to the cancer-related cachexia. So without further ado, let's look at that today. So let's move on to the next paper. This paper was published in April of 2024, and it says the activation of GPR81 by lactate drives tumor-induced cachexia. And it says here that cachexia affects 50 to 80% of patients with cancer and accounts for 20% of cancer-related deaths. We saw that in the prior paper. But the underlying mechanisms driving cachexia remains elusive. Here we show that circulating lactate levels positively correlate with the degree of body weight loss in male and female patients suffering from cancer cachexia, as well as in clinically relevant mouse models. Lactate infusions per se is sufficient to trigger a cachectic phenotype in tumor-free mice in a dose-dependent manner, which means that the more lactate they inject in someone, the more cachexia that is seen in these mice, this mouse model that doesn't even have cancer. I hope this is spinning some mental wheels for us. So we know that cancer, because of the Warburg metabolism, is going to secrete high amounts of lactate. Lactate is going to be having an effect directly on the fat through this GPR81, and it's going to lead to energy expenditure and body weight loss. This is just one small piece of the mechanism, but important. The next paper was published in April of 2025, just a couple months ago. MIF-ACKR3 causes irreversible fat loss by impairing adipogenesis in cancer cachexia. And it says here, both exercise and cancer can cause adipose tissue shrinkage or weight loss, fat loss. However, only cancer-associated weight loss, namely cachexia, is characterized by profound adipose inflammation and fibrosis. Here, we identified tumor-secreted macrophage migration inhibitory factor, MIF, as a major driver that skews the differentiation of adipose stem and progenitor cells, ASPCs, towards a pro-inflammatory and pro-fibrogenic direction with reduced adipogenic capacity in cancer cachexia. It says these findings unveil this MIF-ACKR3 signaling as a critical link between tumors and cachexic manifestations, providing a promising therapeutic target for cancer cachexia. And again, it's about the cancer releasing lactate, but also inflammatory cytokines. And again, we talked about that in the prior paper when we talked about inflammation, cytokines, cancer-related cachexia. This is just further defining how it actually happens. Basically, the cancer will secrete certain factors that turn fat into kind of more inflammatory fat that leads to activation of these inflammatory cascades, which leads to further weight loss and cachexia. Now I want to change gear for a second. We've looked at multiple mechanisms, including inflammation, including lactate, but let's look at some other potential mechanisms that are involved here that have not yet been truly talked about in a meaningful way, in my opinion. So this paper was published further back in 2014. It's called Cancer Cachexia, Understanding the Molecular Basis. It says the occurrence of cachexia in cancer patients is dependent on the patient response to tumor progression, including the activation of inflammatory response and energy inefficiency involving the mitochondria. Interestingly, crosstalk between different cell types ultimately seems to result in muscle wasting. And we see here we have a tumor burden, we have alterations in protein and amino acid metabolism. We see that inflammatory mediators are involved, and we see excess apoptosis, decreased regeneration of muscle cells, which leads to loss of skeletal muscle mass and function. And we see here that there's a breakdown of protein, there's decreased protein synthesis, decreased amino acid transport, and these branched chain amino acids are not being used for muscle building, but they're used for oxidation or energy production. The reason I pulled this paper was because of this next graphic here. So we see here that basically another mechanism in which skeletal muscle are basically harvested for amino acids by cancer cells. So you see here that proteins are being broken down in the muscle cells. And these are just two amino acids we're talking about. Alanine is being transported back to the liver, be converted to glucose, similar to that of lactate. Glucose is being made through gluconeogenesis. Glucose is being transported to the tumor and dropped off so that it can grow out of control. Then glutamine is also being harvested from the muscle cells and given directly to the tumors via the bloodstream. So this is the other big piece of it too. It's not just lactate. It's not just these other inflammatory things. It's also the protein degradation and the harvesting of the amino acids from these muscle cells as well, both to make glucose and for the glutaminolysis that is needed for these cancer cells. And then here we have the Cori cycle where we have glucose being converted to pyruvate, pyruvate being converted to lactate, lactate being shuttled back to the liver, gluconeogenesis happening, and then glucose being given to the tumor. Remember, this is an ATP-driven process. This is an ATP-driven process as well. So we're losing energy on these parts to then provide energy for this tumor, right? So it's sucking the life literally out of us from everywhere in our body so that it can build itself. It has no regard for the host itself. It just wants to grow and grow and grow. I felt this was an important graphic to show the complexities here. This paper was published just a couple of months ago, June 2025, and it further talks about, remember in this paper it talked about the gut microbiota and cachexia? Well, the new paper talks about further mechanisms of how that actually works. So this paper was titled Modulating the Gut Muscle Axis, Increasing Short-Chain Fatty Acid Production, Gut Microbiota Commensals, and Decreasing Endotoxin Production to Mitigate Cancer Cachexia. And it says here that gut microbiota dysbiosis, particularly the depletion of short-chain fatty acid producing bacteria, may contribute to the progression of cancer cachexia. Studies in both murine models, mouse, and human consistently associated cachexia with the decline in short-chain fatty acid produced in the gut microbiota commensals and an overgrowth of pro-inflammatory pathobionts, basically pathogenic bacteria or an imbalance of good bacteria and bad bacteria. These microbiota imbalances may lead to the reduced levels of short-chain fatty acids and branched-chain amino acids and alter the normal bile acid profile. BCAAs and the maintenance of normal bile acid profile are associated with muscle synthesis and decreased breakdown, while short-chain fatty acids acetate, propionate, butyrate contribute to the intestinal barrier integrity and immune regulation. Short-chain fatty acid depletion may increase gut permeability, allowing bacterial endotoxins such as LPS or lipopolysaccharide to enter the bloodstream. This leads to chronic systemic inflammation, muscle catabolism, and impairment of anabolic pathways. So now we're starting to see that there are probably multiple ways that systemic inflammation is being created, not only from the tumor itself secreting inflammatory cytokines, not only because of a disorganized immune system, but also because of the imbalances of good bacteria, bad bacteria, ones that produce short-chain fatty acids have an anti-inflammatory effect, or ones that have a pro-inflammatory effect. And then because of the lack of short-chain fatty acids, then you start to have basically gut permeability or basically a leaky gut syndrome, which leads to further uptake of these bacterial endotoxins such as LPS, which leads to a further inflammation. And then round and round this cycle goes. So you can see that there are multiple systemic vicious cycles that are going on to accelerate this cancer cachexia process. I thought that was pretty interesting. And then it also has a great graphic as well. So we have, again, muscle mass. Here it is, muscle cells. It's releasing glutamine. Glutamine is directly participating in mitochondrial substrate level phosphorylation for energy production, for redox homeostasis, for biomass, et cetera, right? But it's being harvested from the muscle mass itself. These non-essential fatty acids can be used to build other stuff, but they're not being used for energy production. And it says here that cachexia basically feeds tumor progression, which I thought was extremely interesting. And it shows exactly why when you exercise and you give it a stimulus, you give your muscles a stimulus to maintain, get stronger, and grow, it basically will create signals in the muscle mass itself that will act. It won't probably outcompete the tumor, but it gives the tumor an adversary to compete against. So that way there's less glutamine available and the muscle is less likely to break down and you're less likely to lose function. And it says here that, like MSO treatment, acute exercise in the form of 45-minute swimming intervention significantly reduced glutamine levels in the serum in both tumor-free and tumor-bearing mice. The reduction of serum glutamine was 30 to 40% and lasted for 2 hours into a recovery period that was restored to baseline at 24 hours. So a 45-minute exercise session led to a 30 to 40% decrease in glutamine availability to the bloodstream because it's being taken up by the muscle cells for repair and regeneration. And it took up to 24 hours before it returned back to baseline, which I thought was pretty cool. In continuation, we found that long-term training in the form of voluntary wheel spinning, this is a mice study, significantly reduced the growth of both tumor cell lines. And after the acute swimming intervention, we hardly observed any difference in gene expressions in the tumor, whereas transcriptional adaptations did occur in the muscle tissue. So basically, like the mTOR and the muscle building process that leads to increased protein synthesis and growth and metabolism was seen after exercise in the normal tissue, in the muscle tissue, but not in the tumor tissue. It was unchanged, basically. I wanted to just take a second here because it mentioned this MSO here. And I'm like, what is MSO? I never heard that before. So MSO is an irreversible inhibitor of glutamine synthetase and it effectively inhibits glutamine synthetase activity. However, MSO is not applicable in clinical practice due to its main effects of causing basically seizures. It has been reported that MSO-induced seizures is alleviated with methionine treatment. I thought that was interesting. But basically what it's saying here is that MSO is blocking glutamate from being converted into glutamine and then being allowed to be used for nucleotide synthesis. I was thinking, could this be something that could be used for metabolic therapy? But it seems like it's probably not going to be an option for us as it has a high risk of having seizures, which I don't think any of us want. We also know that exercise can significantly decrease blood glucose acutely and chronically. So, for example, if you were to eat a meal and you were to look on a continuous blood glucose monitor of what happens to your glucose after that meal, you see a spike here. If you were to exercise within 30 minutes of that meal for a period of 30 minutes, and that could just be walking, you can see a giant reduction from 12 millimole all the way into 7 millimole, a nearly 50% reduction in the spike of glucose that you have if you do that exercise after you eat. So not only is the exercise going to restrict the cancer of glutamine by 30 to 40%, but it's also going to restrict the cancer of having glucose by 30 to 40% when you eat a meal. On top of that, when you look at exercise programs and what it looks like in terms of reducing long-term glucose control, we see that if you were to do aerobic exercise, it can lower your A1C by 1.33%. Resistance training can lower your A1C by 0.55%. And when they're combined, it can lower your A1C by 1.74%, which is a huge amount of average blood glucose that is being lowered from exercise. And then in particular, I like rebounding because I have a rebounder in my garage and I like to use it on a regular basis. But there was a study that was done with rebounding and it showed that after 9 weeks of rebounding, 3 days per week, for about 20 to 30 minutes per day, it lowered these patients' A1Cs by 1.53% by itself, which I thought was astounding. I want to finish out this piece on exercise. Next paper was published in January of 2018, titled Cancer and Exercise, Warburg Hypothesis, Tumor Metabolism and High-Intensity Anaerobic Exercise. And it says here that high-intensity anaerobic exercise was shown to inhibit glycolysis, and some studies in animals showed that the effects on tumor growth might be stronger compared to moderate intensity aerobic exercise. High intensity exercise was shown to be safe in patients. However, it has to be applied carefully with an individualized prescription of exercise. So what did these guys actually find? Well, it said the study presented by this group yielded similar results because I've clipped out a very snippet of this paper. And it said the authors suggested that high intensity exercise training may be a viable strategy against tumors. There was a reduction in tumor growth, which was associated with an impairment of tumor cell glucose and glutamine metabolism. Exercise training increased the lifespan of these rats by 2.8-fold or 2.8 times. And the tumor mass related to the whole body mass was significantly reduced by 10% in the trained rats, and was 34% smaller tumor mass compared to the control group that didn't exercise. The high intensity training changed the pattern of glucose and glutamine metabolism presented by these Walker 256 tumor cells isolated from animals, whereas exercise reduced lactate production by 72% and glutamine decarboxylation or glutaminolysis by 98.2%. The metabolic changes were accompanied by a reduction in symptoms associated with cachexia, such as excessive body weight reduction and decrease in food intake. So this is astounding. Basically, high intensity interval training or high intensity exercise both steals glucose away from the tumors as well as glutamine. But because it does that, it actually decreases the amount of lactate produced by cancer cells up to 72% and the amount of glutamine used by tumors by 98.2%. And because they're exercising, the excess body weight loss was reduced. And because they were exercising, they had increases in appetite and an increased food intake, which overall helped kind of mitigate the cancer cachexia or even reverse it. Now, what I thought was probably the one of more impressive hypotheses of how this is actually happening. Why is glycolysis reduced by 72% and glutaminolysis reduced by 98%? Why is that? Well, the reason why they hypothesize that is because when there is no lactate being produced from exercise, there is a relatively low amount of blood lactate in the systemic circulation. So when these cancer cells are hyperproducing lactic acid because they're burning glucose like crazy, they produce the lactic acid, and then it's being easily transported into the blood vessels and then either taken up by the liver and converted back to glucose or contributing to the tumor microenvironment and inhibiting the immune system, et cetera, because the gradient is low. However, because of high intensity exercise and because of the lactate that you're creating in the systemic circulation, they believe it actually creates an increased gradient. And so the lactic acid that normally would easily get out of the cancer cells passively is now not able to. So therefore, the lactic acid production that gets built up inhibits glycolysis because the pH inside the cell drops, and that inhibits a glycolytic and likely the glutamine related enzymes as well, therefore inhibiting both of those processes. So I've been asked in a variety of settings, in courses and consults, on the comments of the videos, is high intensity exercise bad if I have cancer? Is the lactic acid that is being created from the muscle cells going to hurt me somehow? And I think that between the fact that we know that exercise in general is going to help steal glutamine away from tumors, it's going to help steal glucose away from tumors on the order of like 30 to 40% in terms of glutamine and upwards of 50% for decreases in blood sugar spikes and significant reductions in A1C and overall glycemic control. Exercise, I believe, is a net win. Not only that, exercise is associated with decreased cancer-related mortality as well. But because exercise creates a demand for glutamine, for example, and other amino acids so that it can maintain the muscle strength and size, and it leads to diverting glutamine away from cancer, I'm wondering now, this is just me speaking out loud on the fly here, I'm now wondering if in addition to fasting, in addition to supplemental ketones, in addition to supplements or medications that block glucose and glutamine before an oxidative stress event like HBOT or IV vitamin C, I'm now wondering if some high intensity interval training or at least some degree of exercise prior to that event to maximally lower blood glucose isn't a bad idea. Only time will tell. In conclusions, it says, as high intensity anaerobic exercise is known to inhibit glycolysis even at distant sites because of the lactate producing muscles, this physiologic principle may be applied as a therapy option to counteract tumor glycolysis. Some rare studies have already applied anaerobic exercise, showing a favorable outcome compared to moderate exercise controls. I would say that one of the big questions that I get a lot is that if I do high intensity exercise, especially anaerobic exercises like weightlifting or sprinting or very intense exercises, do these exercises and the lactate that is made from those exercises, does that harm me or does that help me? And I think that today's video in particular has hopefully put to bed the possibility of thinking that this intense exercise related lactate is going to have a negative effect. It seems to actually have a very protective effect because of what we just saw in the graphics related to the concentration gradient that the cancer cells will now have to push against when there's lactate in the blood already from the exercise. Not to mention, we looked at how intense exercise can lower blood glucose and blood glutamine levels to kind of shunt those resources away from the cancer cells and make them less likely to be fueling the cancer. If you like videos like this, please like, share, and subscribe. And until next time.